Hello again. It's our third meeting. My name is Sергeyeva Жанна, and we are talking about plasmids, their diversity and necessity in nature for bacteria and also for human needs, like using them in biotechnology. So, if we want to use plasmids in biotechnology, we must know something about their properties, and we already talked about their size, their copy number, incompatibility of plasmids, or the possibility of plasmids, of two plasmids or even more plasmids existing within one cell. So we talked about all these things, and now we can analyze this information and understand whether we can use these plasmids as vectors for creating of recombinant organisms. But before we can use plasmids, we must isolate them. It's like before using something, we must get it somewhere. So if we talk about plasmids, yes, they are present in nature, but how can we use them if they are somewhere else? We need to get them somehow for ourselves. We need to isolate them. And so today I would like to talk to you about isolation of plasmids. So you remember that bacterial cells, they have the genome and they have a lot of plasmids, or one copy of plasmid. It can be a mega plasmid, it can be small plasmid. So there are plasmids within bacterial cells. And now if we want to use these plasmids, we must somehow get them. So let's talk about isolation of plasmids. The principle is rather simple. We must break down the cell and take out the DNA, the total DNA, including plasmids. But how we can do it, and if we do it, we must perform it so that we won't ruin the plasmid DNA. And so these methods, there are a lot of actually methods of isolation of plasmids, but they have the main idea they have in common, like breaking down the cells and taking out DNA, separating it from proteins. But these methods differ depending on what species of bacteria we are working with, what's the special properties of these bacteria. It's like how long do they grow, what do they eat, about what are their metabolites. So all these things can influence the isolation of plasmids. But the main idea is the same. We must harvest as many bacterial cells that carry the plasmid as possible. We must break down these cells. Then we must separate the DNA from the rest of the bacterial cells and proteins. And then we must somehow clear this acquired plasmid DNA. If more in details, so first of all, we must collect the bacterial cells. And you remember I told you that if we want to get something, we must get just as many copies of it as possible. And speaking about plasmids, yes, there can be plasmids that are present in, for example, low copy number. It means that we must get as many cells as possible, so it will influence the total amount of plasmid DNA we acquire. But if we talk about high copy number plasmids that are present in bacterial cells in high copy number, so we must make this number even bigger than it is usually in the cell. How can we do it? The easiest way is to put this cell with the genes under pressure, under selective pressure. If the plasmid carries antibiotic-resistant genes, we must put the cell that contains this plasmid on the medium with antibiotic. And the copy number of the plasmid will increase. And we will harvest these cells, and then we must make their concentration as high as possible. We centrifugate them. Then we use lysis buffers to break the cells down. Usually these buffers contain alkalines and surfactants. And then we must part the DNA from proteins and the rest of the cells, and later we just try to one more time clean the DNA of the proteins, because we don't need proteins when we isolate plasmids, but within the cell DNA, proteins are in very complex relationship, because proteins are enzymes and they perform all the actions on the DNA. So within the cell, DNA and proteins are connected. We must separate them just as much as possible. And if making such a conclusion about isolation of plasmid DNA, so first of all, we must make just as more plasmid copies as possible. For example, putting the cells under the selective pressure. Then we must wash off the media with metabolites of these cells where they were growing. We don't need them. We must wash them off. And we have the suspension of our cells. Then we break our cells down. We break them down, we ruin those cells, and the DNA and everything that was within the cell comes out. Then we neutralize all the buffers that we used, so that our DNA won't be influenced by these different buffers. And then we must purify plasmid DNA just using all the possible methods. So after the purification, we have our Eppendorfs, we have the DNA. But do we really have it? Because when we work with bacterial cells. We isolate plasmids, but we cannot, just look at them and see them. It's impossible. You can see something like a little bit of jelly, floating in this tube, but you cannot be sure that the plasmid is there, that it's okay, that it's not broken, that we can use it. So we need some method to visualize our results. And one of the possible methods, and rather good actually, is using electrophoresis for separation and detection of DNA molecules, including plasmid molecules. So let's talk about this method. So method of agarose electrophoresis, gel electrophoresis. Why it has such name, agarose gel electrophoresis? Because usually for separation of DNA molecules, we can use agarose gel. Well, it's just simply a jelly that we use to separate DNA molecules. It has special wells, like holes, where we put our DNA to. The tank for electrophoresis contains buffer, and the main thing is power supply, the electricity that moves DNA molecules through the gel so that they separate. Why do they separate? They separate because they have different sizes. So our plasmids, fragments of DNA, they differ in sizes. The smaller is the molecule, the higher is its speed. And the electricity pushes through the gel, and so the molecule runs quick. If it's large, if it's megaplasmid, for example, it cannot be as quick as small one, and it stays up in the upper part of the gel. So, one more time. Electrophoresis, we have the tank containing buffer, we have the gel. It can be polyacrylamide or agarose gel. Well, usually I worked with agarose gels. So we have wells for our samples. Here is the place where we put our DNAs. We use special loading dye that makes our samples heavier so that they will remain in the wells, not just floating somewhere in the buffer. So using this special dye, we put our DNAs to the wells, and DNA will move from minus to plus because it is negatively charged. So when we turn the power on, our DNAs, they will move through the gel because it contains pores. And the smaller is the molecule, the quicker it will move through the gel, because the big molecule won't be as quick as the small one. And you can see that the smaller DNA runs quicker and faster, and the larger stays in the upper part. It's not that quick. And so the electricity pushes them through the gel, separating them. We can use special markers to find the size of our sample, so we can know that if it's, if for example it is here, so the size probably is like 550 base pairs. Here you can see the photo of the gel with shining samples. They shine because they were dyed with ethidium bromide. It shines orange in ultraviolet, and it makes our results visible. You'll see this is the marker. These are the samples. The thicker they are, the more glow they give. Tells us that there is big amount of samples, so a huge amount of DNA, and we can tell the size of our DNA, we can tell how many DNA we have acquired. We can see that, for example, some samples are empty, so no stripes, no samples. Here we can see the stripes, so it means that we have our sample done, so we can check the size of it, we can analyze our molecules, we can analyze the information acquired about our DNA separation with the help of agarose gel electrophoresis. Well, I believe this is the end of the video. So, different forms of DNA molecules also move with different speed. So, supercoiled form that is actually present in the cell moves with a certain speed. If it's in a circular form, it moves quicker. The linear form moves slower because it cannot go just as quick through the pores as the circular molecule. And here you can see the picture. Here you can see the marker. The marker means that these are the fragments of DNA with the sizes that are already known to us, so we know them. And here are the samples. They are big, beautiful, a lot of DNA. So this one is heavier than the second one, two, three. The second one is heavier than the third because it is situated a little bit higher than the third one. So here you can see one more time the gel with the samples dyed with ethidium bromide. Ethidium bromide is a mutagenic compound. It's rather dangerous because it sort of intercalates, it builds into the DNA molecules. It gives us, when we study DNA molecules, the ability to see them because they shine. But if it is dangerous because it causes mutations. Ethidium bromide can cause mutations in living organisms because it's built into the DNA. So we must be very careful when we work with ethidium bromide. But you can see that the results are beautiful. In the ultraviolet, we see the orange stripes of DNA separated in the agarose gel. Okay, so these are actually the results of my own work. I don't remember whether these were PhD thesis or some sort of article, but anyway, I was comparing 3 different methods of plasmids isolation. Here, well, the quality is not that good, but the plasmids are present here, and different methods that were used, they showed probably the same results. They give the ability to isolate plasmids. And if we compare megaplasmids and smaller plasmids, so the picture will look like this. Well, these stripes, the same for each and every sample, these are the remainings of the chromosome. These plasmid isolation methods are focused on plasmids. The chromosome is much bigger molecule, and these methods are rather rude for it, so it breaks into pieces. And we cannot get rid of these pieces, and we can separate plasmids from these pieces using electrophoresis, but they go all together. Well, these are the pieces of the broken chromosome. All the plasmids that are in the upper part above the chromosome are considered to be megaplasmids. Big plasmids, usually in a low copy number. If we talk about plasmids that are below the remainings of the chromosome, these are small plasmids. Here you can see this is a small plasmid, or middle-sized plasmids. So they are situated under the remainings of the chromosome. So we can already make some sort of analysis of the size of the plasmids that are present in our cells. So after the isolation of plasmids and separation and detection with the help of electrophoresis, we can also use some methods for further cleaning of our samples. We can actually take the plasmid out of the gel because it's absolutely clean here from all the other DNA molecules, RNA remainings, from the proteins, a clear DNA, and we can isolate it from the gel. Now that it is absolutely clean and we know the size of it and we can see this stripe, this DNA, we can use ethanol to precipitate the DNA, and also we use phenol to clean it off the proteins. So we can use just a few more methods to make the DNA even more cleaner than it was already in this method used. And after acquiring that quality of DNA, we can use it for some further goals. And actually we can use plasmid DNAs to create vectors, to create cloning vectors or expression vectors, and actually vectors are the so we are going to talk about vectors in our further meeting, our next meeting. This is actually what I was going to tell you. So as for today, thank you for your attention and goodbye.